Welcome, everybody. Good afternoon, and welcome to the PRISM e-Symposium. I am Dr. Gabriela Sanguinetti, and I will be serving as moderator for this session. Oscar Cifuentes is my co-moderator.
Hi, everyone.
You are on the Biomedical Sciences concurrent session. This session will have two parts, one from 5 o'clock to 5:30 and one from 5:35 to 6 p.m. In this session, we will hear from students presenting on topics related to controlling gene expression and figuring out what makes humans unique. If you have questions, please use the Q&A button below to write them and send them in. And we will select questions at the end of each presentation for our presenters. Next slide, please. Our first presenter is Isaac Paredes, and the title of their presentation is An Evolution in Bioinformatics Exploration of Vitamin B12 Absorption. Isaac?
Thank you. So, as heterotrophs, animals acquire all of their nutrients and energy through the consumption of other organisms, and each animal has a unique repertoire of specific micronutrients that they must obtain from their diet to sustain life. The inability to synthesize some micronutrients places dietary restrictions on organisms. Vitamin B12, also known as cobalamin, cannot be synthesized by any animal, but rather by the cyanobacteria within the gut of most animals. To be absorbed, free cobalamin must bind to a transport protein called a cobalamin intrinsic factor, CBLIF, to form a complex that will then be endocytosed into cells. The intrinsic factor is a glycoprotein secreted by a gastric epithelial cell known as parietal cells. Next slide, please. Cobalamin is a molecule important for DNA synthesis, metabolism of energy and lipids, alongside detoxification. The molecule can also be used as a neurotransmitter and hormone messenger. Overall, it is a very important molecule and an essential dietary vitamin for humans. Next slide, please. Next slide. Thank you. Cyanobacteria in humans are endemic to the large intestine. However, absorption of cobalamin is restricted to the terminal ileal region of the small intestine, rendering most, if not all, of the cyanobacteria synthesized cobalamin unusable. Vitamin B12 begins its journey in the food we eat. Hydrochloric acid and gastric protease free B12 from consumed food. The vitamin then binds to haptocorin for protection from the acidic environment in a complex known as hollow haptocorin. After entering the small intestine, pancreatic protease in the duodenum releases B12 from the hollow haptocorin complex. The freed vitamin then binds to the intrinsic factor for receptor-mediated endocytosis at the terminal end of the ileum. This complex is then broken within the cell, and the B12 is then transported to target tissues via a transport protein in the blood called transcobalamin 2. Next slide, please. Animals obtain vitamin B12 in a plethora of ways. Herbivores in particular may be ruminant or non-ruminant, which further dictates how they obtain their B12. Ruminant species, such as cattle, sheep, and giraffes, obtain B12 directly from their gut cyanobacteria since they regurgitate their food as cud in a cycle of chemical and mechanical breakdown. Non-ruminant species, such as elephants, rhinos, and horses, are seen to obtain their B12 from fecal matter on the forage they consume. Carnivores obtain B12 directly from their diet, which is composed mainly of meat. Meat, for the most part, is actually very rich in the vitamin. Next slide. We hypothesize that the promoter sequence of the CBLIF gene has changed in the human lineage as our vegetarian ancestors shifted to a diet rich in meat, eventually changing towards expression in the ileal region of the gut in humans. Next slide, please. Our first step involved compiling a list of herbivores and carnivores whose full genome sequences have already been available. We then extracted the CBLIF promoter sequence from each of these organisms via a genome browser and annotated the region to highlight the starting site, promoter sequence, introns, and exons, as you can see here. From there, we intend to work on finding sequence motifs among the gene promoters that we collected. Next slide, please. In the coming steps, we will scan the sequences for motifs that are overrepresented within each of the 2 listed organisms, and then compare the results between the lists. The goal of this search is to discover known or possible transcription factor binding sites or other regulatory sequences that consistently correlate with either herbivory or carnivory. We will then search for these regulatory motifs in the human CBLF gene promoter to determine where the human CBLF gene fits among the 2 groups of organisms. The goal of this research is to better understand the evolution of human beings with regard to the genetic mechanisms of our diet. Next slide, please. I would like to thank Dr. Nathan Lents, Dr. Edgardo Sanabria Valentin, and Dr. Gabriela Sanguinetti for continuous support throughout the research process. Thank you for your time.
Thank you, Isaac. I have the following question from the audience. Why are you focusing on the promoter region?
So the promoter region is actually extremely important for the expression of proteins. The promoter region controls RNA polymerase binding to the DNA, and RNA polymerase transcribes the DNA and eventually is translated into a functional protein. So all that really means is that the promoter region controls when and where a particular gene is expressed within an organism.
Thank you. With that, we will move on to our next presenter. Next slide. Our next presenter is Arianna Mahadeo, and the title of their presentation is Effective Exposure to Epigallocatechin 3 Gallate and Epicatechin in the Proliferation of Breast Cancer Cell Line, MDA-MB 468. Arianna, next slide.
Thank you. More than 200,000 women in the US are diagnosed with breast cancer each year, making it the most commonly diagnosed cancer among women. While we may not understand the exact biology of breast cancer, diet is considered an important factor in breast cancer prevention. Catechins are chemicals found in different foods and beverages as shown in Table 1, and these compounds are believed to have potential health benefits, which include chemopreventative properties. The table on the right, which you can see, shows some of the foods that contain high levels of catechins, which are blackberries, red wine, and green tea. Green tea has actually been linked to a decrease in breast cancer risk due to the presence of catechins. Epigallocatechin 3 gallate has been frequently studied in relation to breast cancer, but less research has been done on epicatechin. Next slide, please. The goal of my research project is to determine the effect of epigallocatechin 3 gallate and epicatechin on the proliferation of breast cancer cells MDA-MB 468. These cells are triple negative, making this type of breast cancer very aggressive with not many specific treatments. Next slide, please. So in order to study the effect these catechins have on breast cancer cells, I grew the cells in DMEM media, and they were then plated on a 96 well plate. The next day, the cells were then treated with catechins epigallocatechin 3 gallate and epicatechin at various concentrations ranging from 1 micromolar to 1,000 micromolars. The cells were then incubated for either 24 or 48 hours, and then an MTT assay was conducted to determine cell survival after being exposed to these catechins. Using the data I obtained from the plate reader, I conducted statistical analysis and derived the following results. Next slide, please. So here you can see survival curves for epigallocatechin 3 gallate, which is the light pink, and epicatechin, which is the purple, at both 24 and 48 hours. So as the concentration of the catechins increases, the survival of the cells decreases. This reduction is greater at 48 hours for both catechins. However, epigallocatechin 3 gallate, which again is the pink, decreases cell survival at any time point, whether 24 or 48 hours. Next slide, please. I then compared the proliferation of cells at extreme concentrations with 1 micromolar being a low concentration and 500 micromolars being a high concentration. And a significant effect is observed when the cells were treated with 500 micromolars of Epigalo-Catechin 3 gallate than 1 micromolar. And as the exposure time increased to 48 hours, this effect was even more pronounced. And you can see this because when the two values connected by the bracket were compared, a p-value of greater than 0.01 was calculated. However, there was no significant decrease observed for Epicatechin. Next slide, please. I then compared the two time points, 48 hours, 24 hours and 48 hours. And like in the previous slide, exposure to Epigalo-Catechin 3 gallate leads to lower survival than exposure to Epicatechin at either 24 or 48 hours. Next slide, please. So from my research, I can conclude that the toxicity of MDA-MB 468 breast cancer cells to Epigalo-Catechin 3 gallate increases with more exposure time and has a higher toxicity than to Epicatechin. This suggests that Epigalo-Catechin 3 gallate might be a good anti-cancer agent and may be used as possible treatment. Future studies would have to address how Epigalo-Catechin 3 gallate induces cell death, and additional experiments would have to be done to investigate how these catechins affect triple-negative breast cancer cells. Next slide, please. I would like to thank my mentor, Dr. Delgado-Cruzata, for her guidance and mentoring, Tony Ann Bravo for her help in completing experiments, Dr. Chang for the use of her materials, supplies, and providing the cell lines, and lastly, PRISM for allowing me this opportunity to present my research to all you wonderful attendees. Thank you.
Thank you, Arianna. I have the following question from the audience. And for the audience, before I move on, please remember, if you have questions for our presenters, you can use the Q&A button below to write them and send them in. So your question, Arianna, is how does the concentration of Epigalo-Catechin used in your cell survival assay compare to the concentration present in green tea?
Okay, so green tea has catechins 26 milligrams per serving, and Epicatechin is only I'm not sure how much, but it's only extracted from that, and I'm unsure actually of how much of Epigalo-Catechin comes from green tea.
Thank you. Our next presenter is Samantha V, and the title of their presentation is Evolution or Expression of microRNA 3928 in humans and other primates. Next slide.
So despite the vast similarities we see between humans and other primates, humans are still remarkably distinct from primate species. But how did we get to this point? The genetics of human uniqueness is still mostly a mystery. Current hypotheses suggest that human-specific features arise from changes in gene expression, and many genetic differences that we see between species are due to changes in gene promoters and other regulatory factors such as microRNAs. MicroRNAs are small, non-coding, single-stranded RNA molecules that function in the post-transcriptional regulation of gene expression. Because of their compact size and ability to rapidly diversify, microRNA genes can evolve easily and quickly from genomic sources and can have far-ranging effects. These characteristics of microRNAs likely contributed to their widespread adoption throughout the evolution of increasing complexity in plants and animals, and their novel emergence can further accelerate the evolution of species-specific gene expression. Prior studies have already shown how the evolution of human cognition is potentially driven by expression changes of microRNAs. However, there's still much we don't know about the genetic basis of human uniqueness, and continuing to identify human-specific microRNAs and uncovering their functions may shed light on a key mechanism of human evolution. Next slide, please. So how does my research fit into all of this? Our goal was to explore the functional divergence of the human-specific microRNA and see how it influences gene expression. To accomplish that, we first identified a human-specific microRNA. We then analyzed the genomic similarities and differences of that microRNA between humans and primates, and then we identified potential gene targets of that microRNA. Next slide, please. To identify a human-specific microRNA, we generated a list of all microRNAs on human chromosome 22. From this list, we used the comparative genomics function of the Ensemble Genome Database to identify MIR3928 as human-specific, which we chose as the focus of our study. Next slide, please. We performed a multiple sequence alignment of MIR3928 stem loop sequence in humans and other great apes. The columns with stars indicate an entirely conserved column, and as you can see, large portions of the stem loop sequence aren't conserved, indicating a rapid evolution of the target genes of this microRNA. Next slide, please. Shown here, we built phylogenetic trees of MIR3928 in humans and other primate species based on sequence similarity. On the left is a tree for MIR3928 with just the stem loop sequence, and on the right is the tree for MIR3928 with the stem loop sequence plus 200 base pairs upstream and downstream. The fact that these two trees differ so much argues that the stem loop has been under intense selection, much more so than the surrounding DNA sequence. These trees also diverge substantially from the accepted evolutionary relationships among these species, shown here. Next slide, please. Which shows that this gene has been under intense selection and has been evolving rapidly, especially in humans. Next slide, please. We also compiled a list of predicted gene targets of MIR3928 from 3 different online tools for microRNA target prediction. These 23 genes appeared across all lists as potential gene targets. We took various combinations of genes from this list and ran gene ontology analysis using the Panther database, which profiles large sets of genes and their known functions. Out of these 23 genes, the 8 highlighted genes were associated with the developmental process of anatomical structures. This represents a substantial enrichment of this particular function with a p-value of 0.04. One of these genes, MAPK1, is known to function in trachea formation, morphogenesis, and development, so we decided to focus on this gene a bit more. Next slide, please. MAPK1 encodes a member of the kinase family, of the MAP kinase family, which is involved in cellular processes such as proliferation and differentiation. Previous studies have shown that the proper signaling of the MAPK pathway is required in the development of the respiratory tract. Gene ontology analysis of the MIR3928 target specifically implicated both the MAPK1 gene itself and the formation and development of the trachea more broadly. So how is this relevant to human evolution? One of the most distinct features of human beings is our development of speech and language, and differences in vocal production between humans and primates can be directly attributed to anatomical changes throughout evolution. One of these changes is in our larynx, which is in the upper portion of the trachea. The position of the larynx in humans is significantly lower than where it's found in other apes. Coupled with our capacity for faster jaw and tongue movement, humans have much more range when vocalizing due to the low placement of our larynx. Its evolution in the human fossil record marks the possible origins of language in early hominins, leading us to hypothesize that MIR3928 may be connected to the evolution of human speech. Next slide, please. To conclude, we identified MIR3928 as a human-specific microRNA and showed that it has undergone rapid evolution, especially in the human lineage. We also identified MAPK1 as a gene target of MIR3928 and hypothesized that it may play a role in the evolution of human speech and language. Taken together, this suggests the potential role of MIR3928 in the evolution of human-specific phenotypes. Our next steps with this project would be to confirm that our predicted target genes are regulated by MIR3928, which can be accomplished through the overexpression and knockdown of MIR3928 in human cell lines. Next slide, please. And I would like to thank the PRISM office and members of the Lents Johnson Lab for making this research possible. Thank you for your time.
Thank you, Samantha. As a reminder for the audience, if you have questions for our presenters, you can use the Q&A button below to write them and send them in. Samantha, I have a question from the audience. Why did you select chromosome 22 for your experiments?
Members of the Lents Johnson Lab were already looking at potential human-specific microRNAs on human chromosome 21, so we decided to also look at human chromosome 22 to see if there were also any human-specific microRNAs.
Thank you. Our next presenter, next slide. Our next presenter is Chanté Guy, and the title of their presentation is Catechin Gallate Modulate microRNA 182 3P in breast cancer. Next slide.
Thank you, Dr. Sanguinetti. Did you know that 1 in 8 women in the United States will develop breast cancer? In fact, every 2 minutes, a woman is diagnosed with breast cancer. Research has linked green tea consumption with a reduced risk of breast cancer, suggesting that this may be due to compounds present in the tea called catechins. These are seen here in figure 1 on the right. So what then are catechins? These are polyphenolic compounds that are studied because of their anti-cancer properties. Next slide. Biological mechanisms in breast cancer are mediated by small non-coding RNA molecules called microRNAs. These microRNAs regulate gene expression by base pairing to the 3' untranslated region of target messenger RNAs. Oncogenic microRNAs have higher levels in cancer cells than in normal cells. One example is microRNA 182 3P, which promotes metastasis and proliferation in breast cancer. Next slide. Data has shown that microRNA levels are regulated by catechins. Figure 3 on the right shows mechanisms by which these catechins regulate microRNA levels. Other studies have shown that oncogenic microRNAs are downregulated by these chemicals. Next slide. As a result, our goal here investigates whether 3 catechins abundant in green tea, namely Epigallocatechin 3 gallate, catechin gallate, and catechin hydrate, affect expression levels of microRNA 182 3P in a triple negative breast cancer cell line, and whether we could predict that this happens through direct interactions between these chemicals and this microRNA. It is important to note that triple negative breast cancer cells are the most aggressive form of breast cancer, and that there are very few treatments for this type of cancer due to the absence of the 3 hormones targeted during hormonal therapy. These research questions have not been investigated before. Next slide. So we carried out 2 experiments. In one, we determined microRNA 182 3P changes of expression, and in the other, we investigated whether catechins could bind this microRNA. MDA-MB 468 cells, which is the cell line that I used, were exposed to 2.5 micromolar of each catechin for 24 and 48 hours, and this was followed by total RNA extraction. Reverse transcription was then performed to synthesize complementary DNA, and microRNA 182 3P was quantified using quantitative polymerase chain reaction. The relative expression of microRNA 182 3P was then analyzed using the delta-delta CT method. Next slide. We also carried out a bioinformatic analysis to generate 2D and 3D structures of these catechins and of microRNA 182 3P. And molecular docking software was used to predict possible binding cavities for these catechins in this microRNA structure. Next slide. Shown here in figure B, comparing the levels of microRNA 182 to the untreated cells used as control, no effect on the relative expression is seen after 24-hour exposure to Epigallocatechin 3 gallate, seen here in red, and catechin gallate, seen in green. We, however, see a decrease in the levels with catechin gallate after 48 hours. This is represented in figure A on the left, which shows the effect of all 3 catechins. Comparing the levels of all 3 catechins to the control, seen here in the first bar on figure A, catechin gallate has the lowest levels. Next slide. This slide shows the structures of microRNA 182 and how these catechins may possibly interact with it. Figure D on the left shows the 3 cavities, 1, 2, and 3 in microRNA 182, where these chemicals are predicted to bind. Looking at figures B, D, and F on the right, it shows these chemicals, which are the yellow structures you're seeing here, interact with this microRNA. Based on the SF5 score, catechin gallate is predicted to bind the strongest to microRNA 182. It has a score of negative 30.5060 kilocalories per mole, which is the lowest of the 3 catechins. Next slide. To summarize, the data suggests that catechin gallate decreases microRNA 182 3P levels, and that its SF5 score predicts it to bind the strongest to this microRNA. This suggests that direct interaction of these catechins to this microRNA is how its levels are modulated in breast cancer, which has never been done before. For future research, we will conduct more experiments with these catechins at different concentrations to better understand how they modulate microRNA 182,3P levels. And the important nucleotides involved in the binding of this microRNA will be investigated by mutational analysis. Next slide. I would like to thank my mentor, Dr. Delgado-Cruzata, for mentoring me and guiding me through this project. Thank you to Dr. Yoel Rodriguez, Fatimata Kafando, and Niende back home of Hostas Community College for their contributions to the bioinformatic analysis portion of the study, to Cassie Campbell and Tolian Bravo for their help in completing the experiments, and thank you to Dr. Chang for providing the cell lines I used. Thank you to PRISM for supporting and funding my project and allowing me to present my work to all of you. Thank you for your time.
Thank you, Chanté. As a reminder for the audience, if you have any questions for presenters, you can use the Q&A button below to write them and send them in. Chanté, I have a question from the audience. Chanté, I have a question from the audience.
Okay.
Yes.
Okay.
How will different catechin structures affect their activity?
So great question. If you go back to my slide 4, or rather slide 6, I'm sorry, slide 6, you see that the catechins have different structures and they interact with the microRNA by different nucleotides. So we predict that these catechins bind to microRNA, but we're not 100% sure if they bind. So what we want to do is we want to confirm that it binds, and whether if it's the binding of these catechins to this microRNA that affects how it modulates the levels of microRNA 182. And what nucleotides are important. So what nucleotides on the microRNA are these catechins binding to that affect the levels? Does that answer your question?
Yes. Yes. And thank you, Chanté. And with that, we conclude our first session. I would like to thank our presenters for their work and ask everyone in the audience to send them a big round of applause. Our next biomedical sciences session starts at 5:35 p.m. in the same room. You can stay here until it starts. But if you would like to check out a different session, you can close this window and go to our website, www.jjay.cuny.edu/PRISM, and follow the links to join a different session. Thank you.